What we're going to do today is talk about this global problem of emerging infectious disease. Something dramatic is going on in the world, and it seems to be increasing in frequency and severity. And I'm talking about what we call emerging infectious diseases. Traditionally, these are defined as diseases that have newly appeared in science. We've never seen them before, or they've appeared in a human population for the first time. They can also be categorized as infections that we know about, but they're increasing in their incidence or in their geographic range. So we're seeing things from Asia suddenly in the middle of Kansas. These usually have an identifiable source. Most of these are animal viruses that have jumped from animals into humans. We call this viral traffic, when viruses shuttle back and forth between species. And what they tend to do as they shuttle is they tend to have mutations, and they tend to become more adaptable to a given host. There has been an alarming increase in the things we've never seen before over the last 30 years. This is a study by the University of Edinburgh. There are 1,417 known human infectious pathogens now. 219 of these are viruses, 538 are species of bacteria, 307 fungi, 66 protozoa, and 287 helminths or worms. A good fraction of these are zoonotic. That means they're animal diseases that jump to humans. This is a graph I made sort of showing back from 1951 the general pattern that we've been seeing, 1 to maybe 3 a year. And then suddenly around 2014, we've started this tremendous increase in brand new things. We're halfway through 2016 at the moment, and we're clear on track to probably have this to be a record year. From January to July, we've had 469 cases of a still unknown hemorrhagic fever, with 120 deaths in Darfur and Sudan. A dengue fever outbreak earlier on in the year in Hawaii. The governor there wouldn't declare an emergency unless the outbreak spread to other islands. And 4 weeks later, a baby was born with brain damage from the Zika virus in Oahu. A new species of Lyme disease, bacteria causing Lyme disease. This one is a lot more severe than the usual one we've been seeing. It's also transmitted by ticks. The Zika virus we'll touch on in a little bit. There's an active epidemic underway in Puerto Rico. Probably 25% of the population will have been exposed to this virus by the end of the year. A new strain of bacteria in Wisconsin and Michigan. It's a soil bacteria. We don't know how it's being transmitted at this time. And then cholera, an ancient scourge. April of this year broke out in Tanzania. So something dramatic is happening in the world. The top part to your left, the world map, shows the emerging disease outbreaks through the period of 1980 for 5 years, and then from 2005 to 2009 on your right. Down below, you can see that the number to your left, actually the number of new viral diseases is increasing, and also the number of infections that are transmitted by insects is showing an increase. So there's a real necessity to understand what's happening here. The risk factors for emerging viral disease, and we're going to keep this to viruses, not bacteria or other pathogens. But the chief thing is an expanding global population. Cutting down natural areas, forests, jungles, ever-expanding human population. Native animal habitats are being fragmented. Their wild food sources are becoming less diverse. On top of this is the worldwide practice of this intensive domestic animal breeding. You've all seen the videos and stuff of how we raise chickens. Just thousands of chickens jammed on top of each other, and it's the same with cows and pigs. What happens is, if these are exposed to wild-type pathogens from their non-domesticated cousins, there's a mixing that goes on with them. And this is not something we see in nature. Animals jam that tight together. So viral traffic is increasing globally. There's been changes in the number of insects and the migration of different animals. Things that serve as reservoirs in nature for these viruses are moving or expanding. Air travel has increased dramatically even since the 1980s. You can be in the middle of the jungle in Borneo and 24 hours later be in JFK International Airport, well within an incubation time of something. The biggest worry is still what we call the RNA viruses. These use ribonucleic acid as their genetic material. And these viruses are very well known to be error-prone. When they replicate, they make a mistake in copying their genetic material, to the point where you might have HIV, but you have, I think the record's 27 strains in one patient. You might have Ebola, but there's 4 or 5 different strains by the time, because it's amplified within you and it's made mistakes. So these are really a big threat. And now, with the increasing number of emerging diseases, we have this problem of syndemics. A good example is human herpes virus 8 and HIV-1. Together, you end up with Kaposi sarcoma. It's a very malignant skin disease. We don't know what's going to happen in areas that have the dengue virus and the Zika virus together. These viruses are related. How do these viruses cross into humans? Well, they can directly jump, although this is fairly rare. A good example is avian influenza. And when they do a direct jump, there hasn't been a lot of adaptation to the virus, to the human. So you might be unfortunate enough to catch this, but you don't transmit it to too many people. There can be an adaptation process. The Nipah virus in Malaysia was derived from fruit bats. So we have one hanging in the tree here. And it passed into the pigs at the slaughterhouses and adapted and amplified, and then it passed on to man. And then you can have this reassortment, which is probably the most dangerous of all. And this is typical of the influenza viruses that go pandemic. There's contributions from wild birds, pigs, and humans, and they'll mix within one of these host species. And they adapt and they spread very well, and this is how you get these tremendous outbreaks of influenza. There's a geopolitical aspect to this. In a fairly recent paper, they looked at 335 emerging disease origins from 1940 to 2004. And they found that there was a non-random pattern of emerging RNA viral disease, and these were associated with what are called biodiversity hotspots. These are high biodiversity areas that are under threat from human enroachment. It's also, this is home to 1.2 billion of the world's poorest people, with some of the fastest population growth on the planet. And what's even more concerning is about 80% of the major armed conflicts between 1950 and 2000 have occurred within these regions. This can act to further spread what would normally be a very small outbreak into something much larger. An example is HIV-1, where the Tanzanian-Ugandan war, when it had occurred, bombshelled the virus out of the area and into the major north-south trucking lanes. And within a decade, the virus had spread all over the continent. We have a lot of concern about what are called the biosafety level 4 viruses. These are highly contagious lethal infections with no vaccine or drug therapy. But we've known how to handle these for well over a century. You identify and isolate suspected cases. You trace all their contacts and isolate them and ensure that they're free of disease. And you perform safe burial practices. Later on, when the outbreak is over, you go back and try to identify and control any vectors that may have been involved in the transmission. 2014 to 2015, the Ebola outbreak in West Africa, none of this was done, and it spun rapidly out of control. The WHO was late in declaring an international emergency. They found they had no resources, no budget. In fact, they were pretty much useless in this. And if it wasn't for Medicine Without Borders, Doctors Without Borders, this would have been much, much worse. Simple basic training was all it took to stop an epidemic of this type. Teaching the locals how to make their own decontamination solutions, how to wear protective equipment, safe burial procedures, how to decontaminate areas, and taking swabs for oral for confirmatory testing. It just requires some manpower and some training. In the USA, we had an overly complex, non-functional bureaucracy directed against our Ebola response. We put the police in charge of it, the DHS, Department of Homeland Security, their Office of Health Affairs, and made public health subject to them. And, well, you saw what happened. The DHS Office of the Inspector General Report 16-18 was very, very critical of what happened. Although the title sounds basically mild, you can get this document off the internet, and I found it to be fairly outrageous. The response was staggeringly inept. They failed to provide adequate protection to the screeners. There was no coordination at any level. There was poor training. Over 100 people entered our borders from West Africa without screening at all. This was compounded by inappropriate guidelines that were initially issued. Conservatively, the federal government spent somewhere around $60 billion, some estimate as much as $120 billion during the past 10 years in planning and preparing for a large-scale infectious disease outbreak. A number of GAO reports, including 2 from this year, show that this effort remains uncoordinated, inadequate, and non-functional. With respect to pandemic flu, pandemic influenza, we still don't have the capacity to rapidly sequence bird flu viruses to look for the mutations. There's 3 genes we think are involved that could allow better spread from person to person. We're not being proactive. Asia remains a major risk area for emerging infectious disease, and there are more people living within this circle than living outside it. We're very worried about influenza A. This virus has its 11 genes separated on 8 different pieces of RNA. And if 2 flu viruses are in the same organism, it shuffles the genes around. We classify these by the proteins that are on their outside. One is called hemagglutinin, the other is called neuraminidase. And there's 16 hemagglutinin proteins that we know of, variants, and 9 neuraminidase protein variants. Normally we only find H1, 2, and 3, and N1 and 2 in humans. This is basically how these are classified. What happens is that in areas where there's a high density of different animals that are subject to influenza, these genes will mix inside. China is a constant worry because over there they have this polyculture, aquaculture, where you have your vegetables, you have your pigsties, and you have your ponds with fish. And it's a unified form of agriculture. It's highly successful, it's highly efficient. The waste from the pigs drops in and feeds the algae in the pond. You have several species of fish there. Some of them live off the algae. You also have ducks and their droppings. And vegetable scraps is fed to the ducks. So it's a whole system here. But you have this tremendous concentration of different species of animals and a lot of viral mixing. And what happens every year is that you'll have something, let's say a bird flu virus from the ducks, and perhaps a human virus or a swine flu virus, and they'll mix in one of the animals, usually in the pigs. The genetic material gets reassorted, and you get something new pop out. And we're having this constant deluge every year of recombinant influenza viruses. That's going on 24/7. Migratory birds, most of these have an avian origin, whether the bird gets sick or not. But migratory birds take these from one pole to the other. The Arctic tern, it's a cute little white bird with a black head, and it spends its whole life flying from the North Pole to the South Pole. It gets to the South Pole, it flies back to the North Pole, North Pole, back to the South Pole. It spreads these through its droppings. This is how you get isolated communities with really severe pandemic influenza. The seasons, November to April, these birds are flying from the northern hemisphere south. April, November, they're flying from the southern hemisphere north. And then around the equatorial areas, it's year-round risk. Why are we so worried about pandemic influenza? It's because of the 1918 flu. This was an H1N1 strain, and its dissemination was a global disaster. The disease spread worldwide within months. Isolated Eskimo villages were wiped out in Alaska. 1 out of 4 U.S. citizens became infected, and 500,000 people in the world died within the space of only a few weeks. The final worldwide death toll was so great that the 1918 influenza pandemic joined the bubonic plagues of Justinian and the Black Death as one of the 3 most rapidly destructive pandemics the world has ever seen. World population is now some 5 times larger than it was in 1918. And this same influenza strain with the same lethality would now kill over 380 million people worldwide. Again, the Government Accountability Office just keeps putting these assessments out. We have really no effective planning for pandemic influenza at this time. U.S. government spent $1.2 billion on Tamiflu, which later studies have suggested it really does absolutely nothing. It shortens the duration of your symptoms by half a day and has absolutely no effect on your transmissibility to someone else or hospital admissions or mortality. But that's irrespective because it all expired last year. These reports are just, they keep coming out. Weeks before the first U.S. Ebola case, Office of Inspector General from the DHS slammed the DHS, HHS, and TSA for ineffective preparedness. This highly promoted National Biosurveillance Center has proved itself almost useless. The decade-long $5.6 billion program called Project BioShield, established by Congress in 2014, is a failure. And later this year, in testimony to Congress, the statement was that biodefense preparations are fragmented and lack strategic oversight without any leadership to provide coordination. This was essentially unchanged from a previous 2011 report. So, in a short term, we've spent $120 billion and really have nothing to show for it. This was kind of overwhelmed by the Ebola outbreak, but during 2014 to 15, there was a large outbreak of avian flu in the United States. And there's the infectious tract there. This spread all over the United States, a tremendous viral traffic. Fortunately, this is not a pathogen for man. More than 10% of the entire U.S. egg supply had been affected by the outbreak, and farmers had to slaughter their hens by the millions. Again, a warning. Another worry is in Asia is this, the Asian live markets. These live markets, Asian culture, they like to eat animals fresh. So you go down to the market in the morning and you pick out your frogs or whatever you're going to eat, and they kill them for you there, and you take them home and eat them that day. What you can't see is in the background here are high residential, this is downtown Kuala Lumpur, and there's high residential, high density living areas right behind the market all over. I think I took this picture 24 hours later. I was lecturing at George Washington University. I could have picked up anything. So this has happened before. In 2003, the severe acute respiratory syndrome, or SARS virus, it came out, apparently I've not eaten these, but apparently these horseshoe bats are very tasty. And this is a virus of horseshoe bats. They secreted it into the environment in low quantity, and it was picked up by the civet cats, which are actually a mongoose type animal. And they're readily infected by this, and they amplify the virus to a very high level, and of course secrete it into the environment, and it jumped into humans. So the end result was over 8,096 cases with 774 deaths. Majority were in Hong Kong. There's a new SARS virus that was discovered this year. It can jump directly from the Chinese horseshoe bats directly to humans. It's highly pathogenic in tissue culture to human epithelial cells. And the original treatments that were developed against the SARS virus don't contain it. So all of this suggests a very serious potential risk of a new SARS-like pandemic that warrants preparedness efforts. But I'm not aware that too much is being done about this. There is some encouraging news. There has been some significant improvements in global alerting. The ability to conduct surveillance and say, look, we've got something unusual going on here, we need some help. And this has been done through the internet. ProMed, Stephen Morris at Columbia University was very instrumental in getting this online. And anyone can subscribe to it. It's free on the internet. You get an average of about 7 reports a day. And whenever anyone runs into anything unusual, they report it. And you can actually follow some of this stuff as it's developing. The Canadian Global Public Health Intelligence Network, this is a data mining system that uses computers and automatic language translation. And they constantly scan the internet and global media sources in 12 different languages. And they had alerted when the original SARS epidemic occurred, they knew several weeks before the WHO announced it that something serious was going on in China because their algorithms picked out the increased sales of antiviral compounds from one of the Chinese pharmaceutical companies in their annual report. And it was already alerted by the time WHO realized anything was going on. EcoHealth Alliance, there's a number of nonprofit foundations that are now working on this problem. If any of you have read any of the highly amusing books by Gerald Durrell, he's an Englishman. When he died, he had started this EcoHealth Alliance. They work in more than 20 countries in Central and South America, the Caribbean, Asia, and Africa. And it's made strategic alliances with the Wildlife Trust Alliance and the Consortium for Conservational Medicine. That in itself is a collaborative effort through Johns Hopkins, University of Wisconsin, and the US GS National Wildlife Health Center. The idea is to stop it here in the wild before it reaches a dense urban area. Can an emerging viral epidemic be predicted? Well, a lot of us think it can. There's some steps for a virus, an emerging virus, to cause an epidemic. Typically, as we've said before, these are animal viruses within a reservoir host living within a defined geographical area, normally in the wild. And they shed a little bit of the virus into the environment, and that keeps their own species sort of at a low grade of infection. And then the step one is the adaptation, where this virus gets established in a second species, depicted here in the green. And the virus has made enough changes to live in this new host, and now it's being amplified and secreted into the environment. This is called niche amplification. And eventually it spills over from this zoonotic pool into humans, and you get these small little micro outbreaks here and over here, and the next year over here. And then eventually you get this human cascade, as we saw with the West African Ebola. And essentially it almost, I mean, it made it across the borders, it made it to England, so essentially it was pandemic, although it was pretty self-limiting. The idea is to try, can we catch it up here, or can we catch it in here when it's still containable and we have time for countermeasures? And a lot of us think we can. Before, it would have been a very daunting, very hugely expensive task. The Rockefeller Institute for years had a program down in South America and other places to look for these new viruses. Zika had been discovered long ago by this program, but funding dried up and things went elsewhere, and we've been left in the dark. As I said, normally it would take a lot of scientists and a lot of effort and a lot of laboratory work, but we've had some new technology come out over the last 10 years. And these are the DNA microarrays. What you have is a silicon chip with hundreds of little bits of DNA on it, photo etched onto there. And You can use polymerase chain reaction and the proper reagents, which are tagged with fluorescent dyes. And you can run it through this machine. And it can look at hundreds of different pathogens at the same time. So what would have taken weeks, if not months, you can now do overnight. It can detect influenza strain patterns, looking for those genes, the 3 genes that we're very worried about in an outbreak. It can also detect drug resistance to the known antivirals that we have against it. It can identify new known bacterial disease agents, emerging hemorrhagic fevers. You can use it you can crunch up the insects and scan them to see what they're carrying. And most importantly, it can detect unknown viruses. And it does this because although more than 20 different virus families have human pathogens in them, there's only 4 families that account for 65% of the viruses that affect humans and more than half of all the known emerging infectious diseases. So by putting some of their genes onto this chip, we can get a partial match and identify the virus family. So this is incredible technology, really. The photomultipliers read this. This is the probes have been washed over this chip, and it's pulled out a number of genes, which are giving you an idea of the family of the pathogens in there. It works on blood, stool, saliva, cerebrospinal fluid, crushed insect samples, soil, vegetation, or just environmental swabs. And it now makes this, the new machine is about the size of a bread box. It's a little bit sensitive, the photomultipliers. You know, you have to handle it with a little bit of caution still. But it now makes it feasible to go into these areas. There's an area in one part of the world called the Kalimantan River, which is in Borneo. And this is a natural laboratory for the study of emerging disease. We have fragmented natural habitats from the palm oil plantations. We have virgin jungle. And we have protected environmental areas for the primates. This is the river. This is a protected reserve. It abuts on virgin jungle. And right on this side is a palm oil plantation. In fact, that's the boom where the ship comes in to load the palm oil on. This is the area. It's British North Borneo. This is the area showing the reserve, the virgin natural forest, the primate reserves, and the green area is virgin natural forest. And this looks to be like a disaster waiting to happen. It's one of only 2 known places on the planet where 10 primate species can be found together, and they're overcrowded. I've never seen so many monkeys in one place before. Extremely high biodiversity. There's also 27 caves that have been mapped, and a good population of these fruit bats, the ones that have been found to carry some of the more nastier viral RNA diseases. Civet cats are there. These are pygmy elephants. They're unique to this part of the world. Birds, snakes, everything, and bordering right up against it are the palm oil plantations with moderate density habitation. And these monkey populations are in here. In some places there's ropes across the river so the orangutans can cross. And it's a bit notable here because the orangutans, there was a paper out several years ago showing they had antibodies to the Ebola virus. Now, if it had been from the Philippines, the Reston strain, that's understandable, that's not pathogenic for man. But it was the African strains. And the paper got a lot of, can we believe this or not? And then EcoHealth Alliance sent a scientific team into Bangladesh, and they found the African strains of the virus there. So this Ebola and Marburg may have a much wider geographic range than anyone ever thought possible before. Well, how to get in there, and you can't just go there if you're going to look for an emerging outbreak. You have to go stay there for a while. And you need to know what's there to begin with. So when you see something new, it's recognizable. So the idea was to get a laboratory and put everything on the laboratory and take it in there, and then make all these memorandums of understanding with people that are already there. And we were getting ready to do this, and then you discuss it among your colleagues. And the idea was, well, why do we have to have the lab on board the riverboat? What if we could set it up on land and inflate it? And then put it on something. This is a boat we're looking at. It's been around the world before. It was used by the whaling people, most recently after the Gulf oil spill. They went out and darted whales and got blubber samples, and those poor animals are just poisoned half to death with heavy metals and benzene and everything. So this is what we're looking at now, but we're just about ready to do something here. And when the rainy seasons are over in Florida, we'll be building the first lab and just see how tough this is. This may be a very good compromise. The insect-transmitted RNA viruses remain an increasing worry. As we saw before, they're increasing in number of outbreaks. An example was the West Nile virus back in what was it, 98, 99. Nobody knew it was here. It was a strain from it first isolated in Israel. And birds started dying off, chiefly crows, all over New York City. It got into the exotic bird population at the Bronx Zoo. They started dying off. And this was before the first human cases appeared. This wasn't, they thought the mosquitoes would die that summer or that winter, and they didn't. And here's what happens when you don't contain a mosquito-borne virus properly. The reason this is not all red now is because you have herd immunity in here. Enough people have been exposed to the virus that there's an immunity. But within a decade, it had spread. Then we come to the Zika virus. This is a member of the family Filoviridae. It's related to West Nile and to the dengue virus. Again, it was discovered way back in 1947 in the Zika forest. They took this poor unfortunate monkey, put him in a cage, and set him up in the trees, and took blood samples until they found something new in him. It was named the Zika virus in 1952. It's a single-strand positive sense RNA virus, and it's mosquito transmitted. There's two principal vectors. Whenever you see a mosquito with striped legs, you really want to swat that pretty quickly because that carries dengue and yellow fever and everything else. Aedes aegypti, these insects have become adapted to humans. They're urbanized. They bite during the day. In fact, we have a population in Washington, D.C. that's survived the last 4 winters. The next one is the very aggressive Aedes albopictus, or the Asian tiger mosquito. It arrived here in the U.S. in some tires from Japan that had been brought over to be retreaded. And we're looking now to see if the normal household mosquito might be able to transmit this, like West Nile virus. So anything with stripes on its legs, you want to give that a quick swat. There was an outbreak in '68 in Nigeria, and this virus has developed into 2 clades. There's an Asian strain and an African strain. From 1951 to 81, the infections moved in through Asia and Indonesia, And then in 2007, something happened to the virus, and it appeared to increase in transmissibility. Looking at the genetic sequences, there's some suggestion that a change in its non-structural protein 1 usage may increase the viral replication rate in humans. Well, it started out at Yap Island, spread down into French Polynesia in 2013. Who did really nothing to contain the spread? It was in Fiji around this time. And it was just a question of time that it was coming here. And again, nothing was done. 2014, the outbreak in Brazil, and now secondary cases in the United States. Only 1 out of 5 people exposed to the virus actually develop any symptoms at all. If they do, it's usually a mild 7 to 14-day illness called Zika fever. Begins with a mild headache. You develop this fine maculopapular rash on the trunk and pink eye, a conjunctivitis. And in about 2 days, the rash starts to fade, and within 3 days, the fever's gone. And the rash, a little bit of the rash remains in the conjunctivitis, and that goes away, and that's pretty much it, or so we thought. When you start seeing large numbers of people exposed to this, you start finding other things. In October 2015 to 16, Brazilian health authorities reported more than 3,500 newborn microcephaly cases. This is typical, and now this sudden jump. And some of these infants had severe brain abnormalities and died. In the worst affected regions of Brazil, 1% of newborns were microcephalic. And this is a normal MRI. This is a baby with Zika. And you can see the whole structure of the brain has changed. We now know this virus specifically targets developing nerve cells. There are some other rare complications. 1 out of 4,000 infections, individuals develop what's called the Guillain-Barré syndrome. This is an autoimmune disorder where you attack your own nerves. It can be extremely severe. It moves distally to proximally, and you can end up on a respirator, unable to breathe. A condition called acute cervical or thoracic myelitis. And we've had 1 case in Puerto Rico of immune thrombocytopenic purpura, which is essentially a hemorrhagic fever. A number of, and we know why, but it's your own immune system that destroys your platelets that are essential for clotting. And we see this in dengue viral infections as well. And because it's autoimmune based, it makes us very worried about a vaccine and perhaps the vaccine triggering this. We have vaccines for other flaviviruses, yellow fever, Japanese encephalitis, dengue. But with the exception of yellow fever, the FDA still classifies these as investigational new drugs. They're not for general public use. We probably will have a Zika vaccine pretty quick, but its regulation may take a couple more years. So, the normal municipality response is adult sighting and larva sighting. These are the foggers to try to control the adult mosquito populations. Normally, this uses Duet. It's a synthetic pyrethroid. And for the water, you can drop in these juvenile analog hormones. These are insect growth regulators to block large bodies of water. With the state of affairs of our public health and epidemic preparedness, probably the best thing is individual protection. And if you'll use DEET lotion, and if you're in the woods, wear permethrin-treated clothing, you can pretty well prevent 99.9% of mosquito bites. And it works very well against ticks as well. And then basic source reduction measures around your home, such as reducing the areas of standing water. We are not Puerto Rico on the mainland. We generally live inside screened houses if we don't have air conditioning. And it's just not quite the same as Puerto Rico where things are out of hand. There's a main canal that goes through San Juan that overflooded with the rains. And because they're bankrupt, there was just nothing left. They don't even have an air ambulance service now. They didn't pay them for the last 2 years. So economically, things got out of control. They didn't keep their mosquito measures down. And consequently, it's there. So here's the life cycle of these mosquitoes. The albopictus can lay eggs in the morning dew. There's enough water for that. When it becomes rainy, the eggs hatch, develop into larvae into pupa. And for the pupa to molt into adults, there's a juvenile hormone involved. And that juvenile hormone, if it's present in the water, the pupa won't molt. So if you don't put fish into your ponds and this type of thing, or you can go down to Home Depot, they sell these pellets of juvenile hormone. DEET was developed back in 1944. It's a thin oil applied to the skin or to clothing. It's effective for mosquitoes, ticks, fleas, chiggers, leeches, most of the biting insects. We originally thought it blocked the olfactory signaling on the antenna of the mosquito, but we now know that it's a true repellent. Insects hate the smell of it. They'll fly away. It also has neurological effects on the mosquitoes, in addition to its known olfactory effects. The pyrethroids, these are natural chemicals produced by flowers of the chrysanthemum and dandelion plants. We've known about these for years. These are very potent insect toxins. They prevent the closure of sodium channels in the insect's central nervous system. Their nerves can't repolarize, and the insects become paralyzed and die. You can treat the clothing yourself. You can go on YouTube. They show you how to do it. You can buy this stuff on the internet. The Army's brought out a permethrin-treated uniform. It's good. What happens, you soak your clothing in this, you put it out in the sun to dry, and it covalently cross-links to the material. So you can wash it a number of times and still, when a mosquito touches the clothing, it dies. So if you wear these around, and there's enough of you wearing these around, you'll find your whole mosquito population drops. These mosquitoes are normally territorial within about 300 feet, unless the wind blows them somewhere else. So around your house and this type of thing, you can actually get rid of your own population. Again, you can go on YouTube and it'll show you can buy this stuff on the internet. It shows you how to dilute it. For pregnant women, we suggest you buy the spray and just spray the outside of your clothing. But the trick is to wear long pants and long sleeve shirts. If you go running around in your shorts and on halter tops, you're going to get bit, unless you wear DEET. Don't use it on the cat. Cats lack an enzyme in their liver that allows them to metabolize this, and they go into seizures and they can die. So you can make these insect traps. In fact, it's kind of fun to go out and look at your collection of dead mosquitoes the next morning. But you take a empty Coke bottle, you cut the top off, and you put in sugar and yeast. Invert the top of the bottle, tape it, put it in a dark place. The yeast undergoes fermentation and it gives off CO2, which is a very, very strong mosquito attractant, and they fly in there and can't get out. So they work very, very well. If you have ticks on your property and you're worried about Lyme disease, And by the way, we've had 300,000 cases since it was first discovered in the United States. You get a little cardboard toilet paper roll, and you soak the cotton in the permethrin, you let it air dry, you stuff the cotton back in there. Because it's not the deer that are carrying it, it's the white-footed mice. And you leave this around logs and, you know, where mice are, and they love cotton, and they'll take it back to their nest and sleep on it, and it's the ticks on the white-footed mice that are carrying the Lyme disease. And you can rid your property of them. So the idea, again, is to stop it here before it arrives here. For the last number of years, we've kind of been working. Resorts don't like to fog or spray things. And the cases we've gotten in the United States generally have been for people down in resort areas in the Caribbean. So the nonprofit has been working closely with one of the Army's insect scientists. And he's come up with this, he calls it ProVector. And these colors here have a purpose because they're mosquito attracting colors. And inside is a bit of something called octanol, and there's some sugar there. And there's a thing called Bacillus thuringiensis, which is a nest, it's dipill, the stuff you put on your roses to stop the caterpillars. It's a spore. The mosquitoes come and they feed on this. Good. And they get the spores in and they germinate and it kills the mosquitoes. And we've tested it in the jungle and it's dropped our mosquito population 93% within 5 days. So we published a month or so ago, we went down to a resort in the Dominican Republic, and we set these things out on the balcony on 2 of the living areas. This is before and this is after. It wiped the mosquitoes out. There's the decrease there and the difference between the treated area and the non-treated area. So they're like a couple bucks. We wanted something that'd be very cheap that even a developing country could afford. And the people that can't afford screens and this type of thing could hang in their homes to break up this transmission pattern. So if you're interested in this type of thing, this is probably the best book I've read on the subject. It's called Spillover, and you can get it off Amazon. Any questions?
I always wondered what happened to the girlfriend of the doctor or the guy that came back from Africa exposed and had the disease. I mean, we just don't hear about that. But
this is a good point because we all thought this was going to be, here we go. Remember RNA viruses. Remember they're constantly mutating. If that had been Ebola Zaire from the original 70s isolate, I don't think we would have seen the same pattern. So Ebola restin is non-pathogenic to man. You can inject it into yourself and you won't even run a temperature. So saying Ebola is not good enough. Saying Marburg virus is not good enough. The Angolan strain they isolated a number of years ago has a 90% mortality. So what I'm worried about is the fact we got away with this with West African Ebola, and we get another strain over here. It may not behave the same. You know, our survival rates for the West African were pretty high. So that doesn't mean it'll be the same with the next strain of Ebola. So that's a real good point.